All right, I think we're all in. Thanks everyone for joining us. Welcome all our participants from the CNMI and Guam. And then if you're here from the American Samoas, welcome as well, or anybody else from around the world who might be joining us today. My name is AJ Barney, and today's presentation will be session 14 on activated sludge. And this will be part 1 on activated sludge. I think, if you've joined us before, you already know me and Valeria as well, are my assistant today, the moderator today. But just quick about me, I'm a research engineer and a certified wastewater operator. And we operate out of the University of New Mexico. We are the Southwest Environmental Finance Center. And specifically, we work out of the UNM School of Engineering. This training is provided by funding from the EPA. They fund us and allow us to do these no-cost trainings as well as our technical assistance efforts. Just a couple housekeeping things. I'm sure many of you are familiar already with the Zoom meeting controls. So you have your mute, unmute button, which I'm not even sure you guys necessarily have access to, but you can type any questions you have into the chat. Valeria will let me know if we have any questions as we go. But feel free to ask questions as we go, anything like that, or reach out to Valeria if you're having any technical difficulties as well. So this series is designed to supplement and reinforce the WPI/ABC need-to-know criteria for the wastewater operator level 1 and 2 exams. Though we will kind of touch on some topics that are that are covered in some of those higher level trainings. So we won't follow the exact list, but a lot of this training is kind of based on that same material and should be a good both field level training as well as certification level training to kind of give us a good operational understanding of activated sludge as well as some of the concepts you'll need to know on your certification exams. So, like I said, this is our webinar series, and this is the 14th session. We are currently here at our Activated Sludge Part 1. I will also be providing the training for Activated Sludge Part 2 in 2 weeks. So we look forward for you to joining us there as well. But here's the schedule that finishes out the year. The material for these trainings can be found at the same website that we've kind of been showing you guys for a couple weeks now, or a couple months. That is our swefc.unm.edu/home or /home/op-cert-trainings/. You can see all our recordings as well as the slides for our presentations. So be sure to sign in if you're attending as a group. The facilitator at your location will go ahead and forward everybody's name to us to make sure that we can get you guys those certification showing that you guys attended, so you can submit them for training hours if you need. So with that, we can begin. The first thing I want to do is kind of start off with a broad introduction to the activated sludge process. And to do that, we'll first ask the question, what is activated sludge? The first thing that activated sludge is it's a secondary treatment process. This is because settling is considered our primary treatment, and activated sludge will typically follow that as a secondary process. So that's why we call it a secondary treatment process. It's also a biological process. This is because the activated sludge process uses living organisms to treat wastewater. More specifically, by using microorganisms, we convert dissolved organic matter and suspended organic matter into settleable solids. This process mimics what we see in the natural environment, with those microorganisms consuming organic material in rivers, streams, lakes. Those microbes are already present in the wastewater, and we are just manipulating and cultivating them in environments that they thrive in to reduce the biological oxygen demand in the form of those organics that are being removed. Which means that like kind of our primary goal of the activated sludge process is to remove the biochemical oxygen demand, which we will refer to as BOD for the rest of the presentation. And that brings us to the how of us creating an environment that those microbes thrive in. So this is done using an aeration tank. So raw primary effluent from settling tanks is introduced to an aeration basin where it mixes with the cultivated biomass that we harness in that ongoing activated treatment process to create a mixed liquor. So this biomass utilizes dissolved oxygen the treatment process provides to consume organics and pollutants, converts them to CO2, microbial mass, and water. So this mixed liquor, after sufficient detention time, will then pass to a settling tank where sludge is settled and can be either returned to the aeration tank to maintain the biomass or wasted to maintain the biomass's health and a correct sludge age. Now, both of these are examples of that ongoing cultivation and manipulation process of our biologicals. Now, what this looks like is kind of a visual interpretation. This is an example of how it looks in a typical activated sludge treatment plant and the typical flow path that wastewater takes. So what we're showing here is how wastewater comes in as plant influent. It's going to pass through pre-treatment to remove some of that trash. And then it goes to that primary treatment that we mentioned earlier to settle out solids, and that produces some primary sludge. Then for the topic that we're covering today, that wastewater passes through to an aeration tank that, like we said, has dissolved oxygen being introduced into it. The wastewater will then pass to a secondary clarifier where you can start settling out the solids that are created and that were already present in the aeration tank. And then you kind of have your step of managing those solids, which is either going to be as return activated sludge that returns to the aeration tank, or waste activated sludge, which is removed entirely from the process along with the primary sludge. So those three concepts of the flow in the aeration tank, the secondary clarifier, and our return activated sludge, those are what is contained in our activated sludge process. So we can kind of think of that, how they're isolated from the rest of our treatment plant when we're referencing our activated sludge process. Though waste activated sludge is important for helping us manage that as well. And like I said, we kind of take that primary sludge and our waste activated sludge, and that's taken to solids handling, where it can either be set where it's sent, where the solid part of that is can be either landfilled or sent as or managed as biosolids for agricultural purposes, and then the water from that's kind of removed from those solids is returned to the front of the plant and also treated. And then, any water that passes the secondary clarifier is then disinfected and discharged as effluent to receiving waters. But I wanted to show this as kind of a visual of where specifically we're talking about today, where that falls in the treatment train, that activated sludge process. Also, broadly, it's important to kind of understand our goals of activated sludge. So, like we said, the primary goal is to remove BOD, and this is because our MPDES permits, our discharge permits, at a minimum typically require the removal of things like TSS and BOD. So that's where our activated sludge comes in, primarily used to remove that BOD. And then if we include our secondary clarifiers as part of the activated sludge process, then activated sludge contributes to the removal of suspended solids as well. So both of those parameters. Now, activated sludge can also be used to remove ammonia efficiently, and with the right orientation can also remove nitrates that result from the ammonia removal as well. Now, the activated sludge process is considered a stabilizing process. So then it will also be removing pathogens in favor of microbes that are less harmful to human health and can aid in many, and then also it can aid in the removal of many other type of pollutants, such as emerging pollutants. Those activated sludge, sometimes the bacteria can consume things like mercury or phosphorus. But primarily it's these other pollutants, and then it kind of just aids the pollutants such as mercury and phosphorus. And typically there is other processes needed to make that an efficient removal. So now that we kind of have a good solid overview of the process, and we've kind of mentioned some of these terms that we're trying to remove in activated sludge, let's talk about those specific components and equipment that we can use to achieve those goals. So activated sludge components, we keep talking about dissolved air, and the way that we introduce that into our activated sludge process is through aeration. We also use mixing to make sure that our biomass comes in constant contact with our organics that we're removing, other pollutants, and our dissolved oxygen. We want to make sure that we have a settling component to remove those solids after it passes through our activated sludge tanks. We have an influent component that is designed to make sure that things are distributed correctly, and that things are being brought into the activated sludge plant at the correct rates, such as RAS and our recirculation. Then there's a couple of, then there's some components that may or may not be part of the activated sludge process, such as our meters that measure our different parameters of our wastewater, sprayers to help settle things like sludge, foam, and also just make sure that what we're discharging is more of a settled product, and then things like recycle pumps, depending on our orientation. So with those components that we'd introduce, I do also want to provide a couple quick terms that we've touched on a little and provide kind of some exact definitions and explanations of what they mean before we go into more details of those activated sludge components. So the first is aerobic, which refers to organisms that grow in and survive in oxygen-rich environments. These types of organisms utilize oxygen in conjunction with consumed organics to produce the energy that they need to carry out their normal activities, and to produce more biomass and reproduce as well. Now, free oxygen refers to the form of oxygen typically that we see in the air when it is not bound to other components, and we call this O2. Now, in wastewater, this is typically measured as the dissolved oxygen concentration, and that represents the oxygen available to microorganisms in the wastewater and provided by the aeration devices or our aeration mechanism. Now, the next term is anaerobic. Now, anaerobic environments refer to environments without free oxygen, but they may have oxygen bound to other components. And that's important for as we're manipulating our flow in the different areas of our basins. Now, also related to oxygen are both biochemical oxygen demand, which is known as BOD, and chemical oxygen demand, which is known as COD. Now, as we've mentioned, BOD is the organic strength of the wastewater. We've mentioned that in previous webinars. And it's the measurement of the amount of oxygen consumed by microorganisms breaking down organics in wastewater. And COD measures something similar. It measures the oxygen required to chemically oxidize the organic pollutants completely in wastewater. Now, we also have the mixed liquor, which we mentioned earlier, and is a combination of the microbes being cultivated in the activated sludge process and the solids present in the activated sludge influent. Then stemming from the mixed liquor is two important operating parameters. The mixed liquor suspended solids, or MLSS for short, is a measurement of the concentration of solids in the mixed liquor. So this is our actual analytical measurement of that concept that we're talking about, of the mixture of microorganisms or biomass and the organics that we're treating. Now, the mixed liquor volatile suspended solids, MLVSS for short, represents that living or volatile portion of the MLSS. Now, both of these variations are used to indicate important operating parameters, such as our sludge age and wasting rate. So keep an eye out for MLSS and MLVSS throughout this presentation, as well as the next presentation as well. So now that we know a couple terms, such as aerobic and dissolved oxygen, that are going to be important for us to understand activated sludge, let's go ahead and talk about our first component that we mentioned earlier as making up our activated sludge process. So this is aeration, and aeration has a couple different purposes. This includes supplying oxygen to our aerobic microbes that use that oxygen to break down the organic matter. Now, aeration also, as a useful byproduct, provides mixing, and it provides some mixing of the contents of the aeration tank to keep the solids in suspension and ensure, like we said, contact between the microbes and the wastewater. And then also the aeration is important for promoting that biological treatment by supporting the growth of those beneficial bacteria, which actually perform the organic breakdown, and then also things like protozoa to help stabilize. Now, aeration is provided through a couple of different means. The first is through diffused aerators, which utilize air that utilizes fine or coarse bubble diffusers that are placed at the bottom of the tank. Now, fine bubbles are created using disc membranes, typically provide better oxygen transfer. And then you also, something to know about our diffusers, that diffusers are more common in modern plants due to that energy efficiency and that uniform mixing. There is also mechanical aeration. These types of aerators use surface aerators such as turbines, mechanical paddles, or brushes that float at the top of reactors to agitate the water and introduce air to our aeration basin. These are typically common in things like sequencing oxygen dishes, lagoon systems, and sequencing batch reactors. They provide oxygen and mixing, but are generally considered less efficient than our diffuse systems. The last way that we tend to see aeration in activated sludge is through jet aeration. This is similar to diffused air in that it pushes air into the system via nozzles that are also typically at the bottom of the reactors. These nozzles are also used to pump influent into the reactor simultaneously with air. This is typically creates a strong mixing along with aeration. It has been seen usually as effective for kind of deep tanks and high oxygen demand situations. These are also, though, considered less energy efficient than things like our diffused aerators. Now, the air is provided by a couple of different means, including both blowers, which utilize like a fan mechanism to blow air from the environment and compress it into our air headers that pushes it along to our diffusers. Then there's also things like compressors, which push air into our aeration basin as well. And these are supplied to our activated sludge reactors through a distribution channel or a distribution system known as headers that provide that air to that diffuser network on the bottom of the reactor. So this is what some of those other systems might look like. So on the left is an example of a jetter system, which has just a couple PVC pipes, which will push it into one single stream into the wastewater. At the top, middle is an aeration blower. On the bottom middle is a surface aerator. And then we have some kind of other blowers on the right, which is a more kind of modern system that's easy to regulate. And, as long as you have good manufacturer operating manuals, can be used effectively and in line with things like our SCADA systems. So that's how those systems work, and that's kind of how we introduce our aeration. Now, I want to give you guys a couple just kind of standard operator procedures that are used for all aeration systems, or that are applicable to most aeration systems. Now, the first that we want to do is with our activated sludge and aeration, we want to make sure to always maintain our DO at the correct concentration levels to ensure that those aerobic biological processes are occurring. And we also want to make sure that we're monitoring the airflow to those diffusers. Now, this is done by maintaining a dissolved oxygen concentration that's measured with a residual of at least 1 to 2 milligrams per liter. Now, each system is going to be a little different for optimal operation, but it's important not to exceed the minimal residual DO concentration by too much. As treatment doesn't increase, if you exceed your kind of optimal level, treatment won't increase. However, your costs for increasing that air output will. Now, the way that we do this is we ensure that we have adequate blower output, correct DO distribution, and we want to make sure that we are putting additional blowers online or shedding blowers when necessary if our system is equipped for that. So that makes sure that we don't have extra blowers running when our DOs are A2 and we don't necessarily need it to treat our wastewater. Now, the airflow itself is controlled by opening and closing the air control valve or control valves, or the output of the blower, by closing the air control valve, or if you have a system where you can increase the output of the blower or the compressors, adjusting that as needed. Now, in either case, that can be automatic or manual. And if it's automatic, it's typically monitored by our SCADA systems and adjusted as needed. So then the other thing we want to go ahead and monitor is our DO concentration. Then the other thing that we take into account is that we monitor our DO concentration using the installed sensors to our systems that are generally inspected regularly, or monitored by our SCADA system. Then we can also monitor our DO using handheld probes and make adjustments as needed with that. Now, our automatic systems will use those installed systems to make adjustments to the control valves using actuators or blower systems if they use a variable frequency drive. But it is also important, even if we have these systems in place, to know what our typical DO levels should be and monitor our controls in case we need to make manual adjustments. Now, it's also important to know that the DO is controlled by either control valves for the air header or an air distribution system, or the output of the blowers compressors, like we mentioned, for those variable drives. Now, blowers and compressors should also be checked regularly to determine the output and make sure it's within the correct design specifications, and that our filters are kept clean and replaced when needed. You want to frequently check the diffuser condition. You want to make sure that the air coming up is in normal operation. It's continuous. We want to have our nice clean bubbles, and you want to also monitor to make sure that there's not malfunctions, which is typically indicated by dead zones, or if you see large bubbles, excessive bubbles bursting through the air surface, which can indicate a possible break in the airline or blown out membranes. So we kind of have our visual observations, we have our analytical observations, and then whenever we identify any issues with this, it's especially with our air, it may be required that we drain the reactors to just above the diffuser or nozzle level to kind of see if we observe any mechanical issues with those abnormalities. Like I said, if you see a bunch of bubbles coming up, you lower the level to see if you see a membrane missing, and that's why it's blowing out air all over the place, and you have giant disruptions on the surface. One other important thing, like the last important thing I want to mention, is that if you have purge lines, you want to make sure to go ahead and purge those frequently to make sure that there's not excessive water or MLSS in your purge lines. That's to keep those clean and make sure you keep getting air to the system. An important aspect of our aerators is to understanding how startup works. So it's going to be different for each blower and compressor system and each different air header and diffuser configurations, but we do have some basic practices that I want to mention. So for your blowers and compressors, you want to be familiar with the manufacturer specifications and your operation requirements. This typically involves knowing things like how to check your pressure differential and flow according to the manufacturer specifications and the values that they recommend. And also being able to make frequent inspections for the air relief valves in your lubrication containers and knowing where those are to make sure that your blower is going to keep running correctly and that's not going to be missing any lubrication that can cause it to overheat. Now, for the air headers, you want to make sure that you're familiar with the air lines so you know that the airflow path is correct. This ensures that you're not losing air or having it run against blocked valves. That can lead to unwanted pressure buildups. And then also during startup for the diffusers, you want to make sure that you observe the air coming out kind of with that 1 foot level above the, or below, with the water being 1 foot above those compressors. You can kind of see that being done on the picture to our right. You can see the diffusers visible just below the surface. So when you're turning this on, you want to make sure that you start an aeration basin at this level where you can observe all the diffusers running correctly before you introduce the flow that you're going to actually be treating. Our next component that I want to talk about is our mixers. Like I mentioned earlier, mixing is essential to keep those solids in suspension, which is important to prevent settling of the biomass and then also ensure uniform distribution of our microorganisms, nutrients, and the oxygen. This helps promote contact between our wastewater and our activated sludge, which creates more opportunities for the microbes in the activated sludge to consume nutrients in wastewater and organics. This also helps mitigate those dead zones that we talked about, where treatment may be inhibited or not occurring at all, which can cause pollutants in those dead zones to pass on without ever being treated or addressed. Now, there are a couple different types of mixers that are used. The first traditional type is a mechanical mixer, which can be installed on the surface or submerged in the reactor. This is an example on our picture on the left. It shows a motor for a mixer. This would be connected to a shaft, which has mixer blades on the bottom, and that keeps the wastewater moving at the bottom. This is especially important for our anaerobic zones and our zones without oxygen for, say, if we're doing some nitrate removal, we can keep them still mixed so treatment is being done uniformly, like we said, and that our biomass is still being moved around and shifted from each area. We also talked about our diffused air, how that can act as mixing. And then there's certain things like our hydraulic mixing, which relies on the design of our reactor to make sure that as our effluence is coming in, our RAS, and if we have any recirculation, the design of the reactor actually allows for those components to mix as influence coming into our activated sludge. The next component that we mentioned is our settling. So after the aeration reactor, settling is a key step in order to separate the treated water from the activated sludge or those biological solids. This takes place in our secondary clarifiers, which are sometimes called settling tanks, or in the aerator reactor itself for some sequencing batch reactors. Now, settling allows for the control of that part of the activated sludge, or sorry, it allows for the control of what part of the activated sludge is returned to the activated sludge process and which sludge is wasted or removed from our treatment train. Now, this also provides an improved effluent quality when those suspended solids are allowed to settle out. So the secondary clarifier where settling occurs are designed the same way as our primary clarifiers, which we presented on a couple weeks ago, and utilizes that same principle of gravity settling. So these work exactly the same. So as long as you have sufficient detention time of 2 to 3 hours, they can remove approximately 99% of your sludge as long as the sludge is managed properly. That means like its age and the level of biomass in there, the amount, the concentration. So these, like I said, just like primary clarifiers, they have an inlet structure that distributes flow evenly and usually has a baffler equipped to minimize turbulence. They have scrapers to collect sludge and move it towards sludge removal ports or suction lines where it can be pumped for recycling or wasting. Finally, they are usually designed with effluent weirs that trap floating solids and allow effluent from the surface of the clarifier to flow to the next stage of the wastewater treatment plant. And then also, there are usually things like control valves to control the process, walkways to access different components and check your characteristics, and then other types of supporting structures. So this is what it looks like from the side, where you can see our V-notch baffles that allow that clean water to be removed, as the rest of our flow allows for some settling. And here's a diagram of what that looks like on the inside. So from the operational standpoint, each system has its kind of own nuances, but unlike aeration systems, operating secondary settler tanks have much more standard processes that we can use and we can recommend, as long as it's kind of that same type of clarifier technology. So the first is to maintain proper flow rates. This helps avoid hydraulic overloading, which can cause poor settling and solids carryover, which is known as short circuiting due to that uneven flow distribution. So that's solids being actually discharged with our wastewater, which defeats the whole purpose of our settling. So as long as we keep our hydraulic rates correct and we're not overloading these devices, we can make sure that we're not sending out the solids that we're trying to trap. Now, also part of that, it's important to monitor our sludge blanket depth. Excessive sludge depth can lead to solids rising and escaping when the effluent, with the effluent, and it can also lead to that short circuiting like we mentioned. Typically checking the sludge blanket is done with a core sampler, which is also known as a sludge judge. Those core samples measure the sludge blanket directly, but the supernate also helps indicate if there's poor settling. So you might see some sheared sludge that is in your supernate, and that can also let you know that maybe your sludge age is too much or other issues like that. Now, the sludge blanket can be reduced by increasing the recycled and wasted sludge rates. And this is kind of the only process where the sludge recycling and wasting rates are adjusted. And this is the main step that you use to manage sludge age, your food to microorganism ratio, and your MLSS concentration. We will discuss this more in our next presentation in 2 weeks. Now, lastly, like many wastewater treatment processes, you want to frequently inspect your mechanical components and regularly check things like scrapers, pumps, and weirs to make sure that they are working properly and solids or growth are not accumulating on them. Then that last component that we kind of talked about, we want to manage is our activated sludge influent. So flow to the activated sludge reactor is either gravity fed or pumped in. Influent mixes with our recycled sludge in this step, either in a designed compartment for mixing that is open to the rest of the reactor or just at the beginning of the reactor when it all comes in together. So on the bottom right, we kind of show some lines coming in. This is typically what it'll look like coming into the front of a basin. You have a main header of your influent and RAS. And then on the top right is a RAS would be a valve for solids coming out of the secondary clarifier, and this is where you can manage your RAS or your WAS that you are removing from the activated sludge process. Now, also in your influent, this is a good point where you can regulate not only the influent, but you can monitor for your BOD or COD to understand if there's any changes or if you're expecting any unusual loads of organic strength. Also at the influent, you may have a anoxic zone. This is where you would introduce your recycled flow and allow for denitrification, which is sometimes introduced in your activated sludge process to remove those nitrates that are created from ammonia conversion. And then the last thing we'll mention, these are all different and don't necessarily have any standard operating procedures, but you often have things like your DO meters, your MLS meters, ammonia, nitrates. This help you monitor the flow and help you make adjustments. You have your recycle pumps, and then also sometimes sprayers, like we said, to kind of get rid of the foam that tends to pop up in our activated sludge processes during changes of influent quality or changes of season. So that is all that's kind of an overview of each individual process. We can go on and talk in much more detail about it, but for our kind of hour long that we have, I think that's pretty in depth. So that brings us to our first poll question. If you'll want to offer that up for Hilaria. So this question reads, so I'm just going to kind of quiz you on some of the stuff we talked about. Which of the following pollutants does activated sludge not remove? And those are, is it ammonia, total suspended solids, BOD, or trash? We'll give you guys about 30 seconds to answer that. All right, Valeria, that's pretty good. We're not getting any more answers, so if you want to show the results. So as you guys can see, we had 78% say trash, which is our correct answer. Activated sludge process does usually remove ammonia, total solids, and BOD. So that's our first question, and we do have one more right away. If you want to pop that up, Valeria. So this next question is, what concentration of DO should be maintained in an aeration basin for optimal activated sludge treatment? The answers you can select are 25 milligrams per liter, 1 to 2 milligrams per liter, 100 to 200 milligrams per liter, and 1 to 2 pounds. If you'll remember, we just mentioned this during our aeration slide. And again, we'll give you guys about 30 seconds. All right. Looks like we got a bunch of answers. So we got 91% selected 1 to 2 milligrams per liter, which is the correct answer. I would like to point out that the last one, kind of had the same numbers, 1 to 2, but it mentions pounds. So it's good that no one selected that. And good job, everyone. So I'll go ahead and end that. And we'll kind of keep going. Good job, everybody. So now that we've talked about those physical components of the activated sludge process, For the next part of our presentation, I do want to discuss the sludge biology and our process variables. So we'll start this section with a discussion of the biological processes that the microbes in the activated sludge reactors undergo. So the first one is aerobic respiration. So this is where our aerobic microbes, along with oxygen and the organic material, like we said, one of the byproducts, just like when we breathe, is carbon dioxide, and then the other is biomass. So they're using, this is like we said, where they're using the oxygen and the organic material to carry out their normal operations, but then also they're producing more biomass and reproducing and kind of just living. Then we also have nitrification. This is where we mentioned we see our ammonia removal. So along with oxygen, microbes will take ammonia, specifically our nitrification bacteria will take ammonia, and with oxygen, they'll create nitrates. And then in our anoxic conditions, some of our facultative bacteria will take nitrates, and along with the organic material, they'll use the oxygen in place of dissolved oxygen to produce nitrogen gas. And then we also have some phosphorus uptake in our microbial processes, where our phosphorus accumulating organisms will store phosphorus that they pull off of different pollutants in the wastewater. Now, we do have a couple of different type of organisms, like we said. We have aerobic and anaerobic and facultative microorganisms. These are all important in our activated sludge process. And in our certifications, it's good to know the difference and what type of activities they may perform. Now, also for our certification and just our everyday operating parameters, it's good to know the different types of microbes that we're going to see in our wastewater. So we have bacteria, which typically make up 90 to 90% of the activated sludge biomass. We have protozoa, which are a little more complicated organisms. We have rotifers, viruses, fungi, and algae as well. These all make up kind of our overall ecosystem of our wastewater. So the first one I'll talk about in detail is our aerobic bacteria. So this is the first one that our operators need to be familiar with. Now, these are the specific microbes that we've talked about that utilize the DO that we've been discussing, and they oxidize the organic pollutants in wastewater, like we've said, for growth and also to increase the overall biomass in the activated sludge reactor. Now, these type of microorganisms provide a majority of the BOD consumption, and they cannot survive without oxygen. So utilizing them along with aeration is really the only way that activated sludge treatment for wastewater is even possible. Now, these organisms are typically single-cell bacteria and are known to reproduce relatively quickly under those favorable aerobic conditions. Now, some aerobes, like we mentioned, are also important in removing ammonia from the wastewater through that nitrification process. The last thing I want to point out about aerobic microbes in wastewater is that they typically have no odor, and so that's the reason. Then after the activated sludge reactor, you don't tend to have those odors, unlike your preceding processes such as pre-treatment, primary, which are usually have more, which don't have aerobic conditions and are much more likely to have offensive odors. So those kind of good aerobic bacteria that we're cultivating in our aerobic reactors don't produce those odors. The next type of microbe that we'll talk about is our facultative anaerobes. These types of organisms will utilize and select for dissolved oxygen when it is available. And I'm sorry, I might have said facultative aerobes, but it's facultative anaerobes, which means that when they need to, they can become anaerobic, which is why, like I said, they select for dissolved oxygen, but can use oxygen bound to other components when that free oxygen isn't available. Now, these types of organisms do prefer your aerobic environments, but are also able to succeed in those anaerobic or anoxic zones as well. They are present in many different wastewater treatment processes, including lagoons and digesters. They can be bacteria or fungi, but the ones that are important in activated sludge are typically our bacteria. And they provide denitrification in some of our activated sludge configurations that utilize recycled sludge or recycled flow. And then the last thing that I'll mention about the facultative anaerobes is that they grow slower in environments lacking oxygen, and these processes can lead to more odors than those aerobic processes. The next microorganism note is our filamentous bacteria. These bacteria grow in long thread-like strands to create mesh-like structures that provide structural support for our activated sludge flocks. These flocks kind of provide a support system that other microorganisms can attach to and effectively remain in contact with the dissolved oxygen and the pollutants that need to be consumed during our wastewater treatment. Now, like with any of our other microbes, filamentous bacteria must be kept in a healthy balance. So excessive filamentous bacteria growth does not interfere with the activated sludge process itself and lead to kind of this foaming that I'm showing you guys on this picture on the right. Now, there are at least 30 different types of filamentous bacteria, and different conditions can lead to the excessive growth of each different species, such as low DO leads to the excessive growth of, I don't remember how to say this one, but Sphaerotilus natans. And then also you have our Nocardia, which is one of the most common ones that we hear, and Microthrix parvicella, which tend to thrive when we have low organic loading and causes foaming under those conditions. But there are lots of conditions that can lead to foaming in our wastewater. And we will talk about those in our next presentation. Now, with BOD, like I said, we also mentioned that the bacteria in activated sludge is also used to remove nitrogen from wastewater. This is done through nitrification and denitrification. Nitrification removes ammonia. Ammonia is present typically in the form of ammonium in wastewater at our kind of lower normal pHs. And then at our normal pHs, but then ammonia is present at our higher, I might have got that backwards. I have to remember how that goes. But in our normal conditions, it's typically present as ammonium. And then ammonia is more toxic. Now, ammonia in our wastewater is a problem because we receive it from, it's all the urine that ends up in our wastewater. And like I said, it is toxic. Then we also have our denitrification process, like I said, that is produced by which is carried out by our facultative anaerobes that removes nitrates. And then our nitrates can lead to methemoglobinemia, or our blue baby syndrome. Now, we utilize a Ludzak-Ettinger process in our activated sludge, which can help recirculate our wastewater, which does recirculate our wastewater to our anoxic zones, where the facultative anaerobes remove nitrate. And you do need to have your denitrification reactors separate from your aeration reactors to carry this out. Some of the specific bacteria that carries this out, the reactions I wanted to show you guys. So for nitrification, we have our nitrosomonas and our nitrobacters, which, like I said, will take ammonia and convert it to nitrite. That's the nitrosomonas. And then our nitrobacters convert that nitrite to nitrate. And then the denitrification takes that nitrate through multiple steps and produces nitrogen gas, which should be N2, not N3. But this is what those chemical reactions look like. Now, there are other organisms that are going to be used as characteristics that we use to kind of understand our characteristics of our wastewater. This can be things like amoeba, which indicate we have a young sludge age. We also have some protozoas, other protozoas such as Giardia and Cryptosporidium, which may lead to waterborne diseases. And then a lot of times we have things like our free swimming and stalk ciliates, which indicate that our sludge age is stable and where it should be. You also have things like rotifers. These are multicellular microbes, and because they're multicellular, that indicates that our sludge age is more advanced. So these are present in that older sludge. And a lot of times these may be associated with that poor settling sludge that we kind of mentioned can be seen in the supernatant of our sludge judges. So that's why it's important just to understand what microorganisms we should be seeing, what we should be looking for, and be able to perform that kind of microscopic analysis. And it's just kind of just showing you a microscope, which is a useful tool for examining our wastewater. So that is the biology component of this presentation. And so that brings us to our next question. This is poll question number 3. If you want to throw that up. So our question is, what type of microbes convert nitrates into nitrogen gas in the activated sludge process? The options are aerobic bacteria, archaeans, facultative bacteria, and nitrosomonas. So the key here is converts nitrate into nitrogen gas. Just a little more time, guys. If you want to just take some guesses, that's okay too. We're going to answer this slowly. It's not a big deal. I'll tell you guys the answer here in a little bit. All right, Valeria, that's probably enough time if you want to show the poll. So most of us got it right. That's good. It is our facultative bacteria. If you remember, they use those nitrates in the absence of dissolved oxygen. So if you remember to convert nitrate into nitrogen gas, that is known as denitrification. It must be separate from our aerobic processes. So it would not be our aerobic bacteria. And our nitrosomonas are important in nitrification, not denitrification. So that's why it's option C, facultative bacteria. And then archaeans are typically present in wastewater and are like our methane producers. So good job, everybody. That brings us to kind of talking about our operational variables. This will kind of be an introduction of that. And then on our next presentation in 2 weeks, we'll go more in depth on our characteristics and what they mean and how to troubleshoot. So the first operational variable I want to discuss is our key parameters that indicate activated sludge efficiency, stability, and compliance. So we want to make sure that we have a good understanding of what these variables are and what they are indicating in our activated sludge process. For anyone who's taken our activated sludge or your activated sludge certification, especially at the higher levels, you'll know how complicated the activated sludge section can be and how much emphasis is placed on it. And it typically has to do, it starts with these key parameters and understanding what each one might mean and what the difference in them may indicate. Now, these are typically our biological, chemical, and physical conditions, which indicate the overall health of our activated sludge. And they can also be determined using lab analysis, our wastewater probes and sensors, calculating other parameters based on our analyses, either probe or lab, and then also just our basic field observations. Now, in order to optimize our sludge activation, like we said, it does require lab analysis and to maintain our detailed records. So that's going to be important throughout operating our activated sludge. So the first couple ones I want to mention, we're going to talk about DO. So like I said, we'll make sure which we've talked about already. And now that you understand the various ways that it's utilized by our aerobic and facultative microbes, I hope it makes a little more sense why we want to monitor the DO concentration in our wastewater. And it's to ensure that those microbes have enough oxygen to carry out their functions. You also already know that BOD and COD is our measurement of the food source for many of our microbes, especially our aerobic organisms that and it's this organic strength that needs to be removed to meet our permit requirements, as well as to protect the health of our receiving waters. It's also important to calculate some of our performance indicators and to make general process adjustments. Now, MLSS, as a reminder, refers to the solids concentration in activated sludge and is used to calculate sludge age, the food to microorganism ratio, and the sludge volume index. Now, MLSS is analyzed the same way we typically measure TSS, which involves filtering wastewater samples and then drying the sample at 103 to 105 degrees Celsius for at least 1 hour. And it can also be measured using a handheld probe or in place of a sensor that reports the MLSS to a SCADA sensor system. A couple more key parameters. We have sludge age. This is calculated using the sludge retention time or minimum cell retention time formula. And the sludge age helps us indicate the overall health for activated sludge and if it's going to be operating optimally to remove our parameters. Alkalinity is also important, or our pH. This helps us understand if there's any toxic loading or any extreme variations which may harm our microorganisms. This is important because a lot of our nitrifiers are known as the divas, or I'm sorry, they're kind of the divas of our wastewater, and they're the ones who are most likely to die when we have kind of adjustments like this or extreme changes to our pH. Alkalinity in relation to pH is important because our alkalinity acts as kind of a buffering agent for our wastewater and protects from those extreme pH changes. And it's also of note because our nitrification decreases our alkalinity and consumes alkalinity as we have more nitrification occurring. And alkalinity can also be a regulatory parameter for our wastewater treatment plants. And then we also want to make sure that we understand phosphorus and nutrients. That can also be, phosphorus and nitrogen, which can be regulatory requirements as well. And those nutrient imbalances can lead to poor settling, bulking, and poor treatment. So there's a couple more parameters that we want to make sure that we are aware of. Now, here's just a kind of extensive list of those parameters: the COD, the BOD, the suspended solids, the dissolved oxygen. And this kind of lists where you might measure them and what those common ranges might be. I want to point out that things like BOD comes in at 150 to 400 milligrams per liter, and typically after an activated sludge treatment plant, it's at 10 to 20 milligrams per liter. So that kind of shows how we get below that typical permit requirement. Same with total suspended solids, comes in high, leaves the plant low, and typically it can leave between that 30-45 rule that we like to mention for our NPDES permits of a monthly average of 30 milligrams per liter and 45 7-day average. Now, these, like I said, are typically measured with a combination of probes or lab analyses. The last one I want to kind of mention that's important to understanding what's coming into our wastewater. We also use it for our different more complicated parameters, like we said, like F to M ratio, is BOD and COD. So just a couple of things about these. Our BOD, like we said, is our biochemical oxygen demand. This is what the wastewater is actually consuming. This is measured in a lab. The most prevalent method is our standard method 5210. An example of that is going on the left right there, where it's measuring the change in DO over time. Well, this is probably instantaneous DO, but it would be a kind of similar setup where you're kind of enclosing that and making sure the DO is not introduced, but see how much it's consumed over that 5-day period while being incubated at 20 degrees Celsius. Now, another way that plants measure their their organic strength is going to be chemical oxygen demand. This is typically measured using standard method 5220, and is actually you use a chemical reagent to force all the organics to be disposed of, and it measures how much oxygen is consumed in that 3 to 4 hour method. This is kind of faster and helps us determine if there's like an overloading in a day, but we don't necessarily use this for those same kind of calculations. So those are just a quick overview of all the parameters I want to talk about. We're going to kind of go over these more in 2 weeks. But with that, it brings us to our last poll question. Brother, if you want to pop that up. So this one reads, which of the following parameters is measured by drying a sample for 103 to 105 degrees Celsius for 1 hour and then weighing the sample afterwards? All right, we'll give you guys a couple more seconds, but it looks like those are the last answers kind of trickling in. Go ahead and show the poll, Valeria. So most of us got this correct. It is MLSS. That is going to be the mixed liquor suspended solids. Typically, this is measured the same way as TSS, which is by drying the sample for an hour at 103 to 105 degrees Celsius and weighing it. BOD, like I said, typically measures the change in DO concentration. MLVSS will actually be a volatile sample, which you heat for much higher and measure the change in weight of the burned off organics. And the total volatile solid will be measured in a similar way. So good job. Most of us got that correctly. So with that, that is my last portion of the presentation. We'll go into more depth about those parameters, the characteristics, and how we troubleshoot those characteristics on our next presentation. But we want to go ahead and open up the floor or give you guys the opportunity to ask any questions if you have any right now. Do we have any questions in the chat, Valeria?
No questions.
Okay. Well, we'll stay on for a couple more minutes, but here's our contact information. As always, reach out to us if you have any questions about certification, training, troubleshooting, anything like that. We are always available. And also feel free to send anything in the chat if you'd like. Other than that, I think we're good for the day, and I hope you guys all have a great day. But like I said, we'll stay on for just another couple minutes if anyone comes up with any last minute questions. All right, Valerie, I think we're good. If you want to, I'll go ahead and